Good afternoon, everyone. Um, I'm going to talk about data processing in Metabolomics, targeted, untargeted, and a little bit about Fluxomics data analysis. So metabolome, as we all know, is the complete set of small molecular weight compounds that we see in a biological system. And metabolomics, as any other omic field, the interpretation and the biological interpretation completely depends upon the identification and correct quantification of the data. So data processing is a very crucial step here. So this has been covered by many other speakers, like why we do targeted versus untargeted, what is important in targeted. Targeted is what we know we are looking for. The analysis is mostly hypothesis driven. So targeted analysis, the main focus is on uh, understanding a biological process rather than searching for biomarkers. So we focus on chemically related compounds, and we just study them in targeted way to see how they are behaving in a particular system. Whereas the untargeted analysis, which is also called global quantification in a system, is more qualitative in approach. So what we try to do here is we look at two different biological states and we compare them. We look at them globally and we try to compare them and see uh, what is changing, what is different in the system, and we try to report the analytes that are different uh, in these states. So targeted metabolomics, these are the common uh, targeted analysis that we do here in our core. These are the common ones, but they are not the only ones. We can also look for something that we have never done before. These are the ones that we routinely do. Total lipid fatty acids, free fatty acid, triacyl glycerol, diacyl glycerol are mostly just GC based techniques, acyl carnitines, acyl CoA ceramides, TCA cycle intermediates are LCMS. Here we do LCM based upon LCMS, and short chain fatty acids and amino acid is we use GCMS techniques to do this analysis. So let's look at the targeted metabolomics workflow. All the targeted metabolomics analysis are done pretty much in the same way. We start by randomizing the sample sequence. The data is acquired using either GC, LCMS, or GCMS, and then we use a software for batch analysis, and we try to integrate and align all the samples, perform calibration and normalization, and the report that you get is in the form of Excel spreadsheet. So let's take an example of amino acid profile analysis. This is the most common analysis that we do here. This amino acid profile analysis is the data is acquired using GCMS. We use a single internal standard, which is Norvaline. We use internal standard at 4 different concentration levels for this analysis for calibration purposes. So this is how, as Charles also showed this before, the Quant Software, Mass Hunter Quant Software. This is a screenshot from the software. And what we do here is after we acquire the data from GCMS, we put all the samples and in this software, all the data files in this software, and then we create a method. And what we do basically here is we focus, so we, for each of these compounds, we plug in their masses and the retention time that we are looking for in the method, and then we plug in the information about the internal standard, the levels, the different levels, the standard, and then the software just we can do it, analyze the whole batch automatically. And once the batch is analyzed, we go back again and we check all these peaks if they're integrated properly, just to make sure about the quality of the data. And if these peaks, they are not integrated properly, we correct them manually. And this, because what we are measuring here is basically the area under this curve, which is this area. And if these peaks are not integrated properly, this results can be different. So this is how we correct it. After this, we generate a data report, which is nothing but a Excel spreadsheet. The information that you see in this Excel spreadsheet is this information that we get from the client when they submit the samples. And so it's basically whatever the ID numbers or the type of treatment that you give. And this is the name of the file, the data file that we acquired from MassSpec, and the time stamp information when it was acquired. And then finally, what you get is these all the amino acid and the final concentration, which are normalized with respect to internal standards. Along with this, as the first sheet, we also, for amino acid analysis, we also give like Excel plots for each of these amino acids, so that you can see for each of these samples, you can see the concentration. For example, like if these are the controls, so you can see the levels here. So just to give the overview of the data, how your data is looking. So this is basically what you get out of a targeted analysis. The report would be mostly an Excel spreadsheet with a plots with the plots in it. Next, let's talk about Fluxomics data processing. Charles already spoke about the Fluxomics part in detail. So Fluxomics, as we all know, refers to the whole set of reaction rates in cell, and this comprises conversion and transportation rates of metabolites. Fluxomics analysis can be seen as another type of targeted analysis because we know what we are looking for. So its workflow is pretty similar to the targeted workflow. We randomize a sample sequence and then perform data acquisition using either LCMS or GCMS. Then we integrate the peak and align them. And as Charles already mentioned, that since in Fluxomic experiment we use C13 labeling, and we also know that naturally this isotope is present around 1.1%. So it's very important to correct for this natural isotope abundance. And we have an in-house software that we use for natural isotope abundance correction. The algorithm was based on the paper published in Journal of Mass Spectrometry by Charles A. Fernandez, and we use the same algorithm for the correction. So after the correction, it's just the report is pretty much the same type of report as you see in amino acid analysis, other targeted analysis. So again, the software that we use for Fluxomics analysis is the same software that we use for targeted quantitative analysis software. And the only difference that you see here is that when we target for the compounds, we target for each of these carbon chain. We target for example, like for malate, we target not only just malate, we target for C13, 1, C13-2, and so on, whatever is the number of carbon, whatever is the number of carbon in each of these compounds. So the method is same. We plug in the masses and the retention time for each of these compounds, and then we go back and manually check each of these peaks to look for integration and make sure the data quality is good. And the Fluxomics report is also similar to the targeted report, where you first see an Excel spreadsheet with all the concentration levels of each of these metabolites. Along with this, we also give these bar plots for where you can see individual labeling in each of these samples. So as you can see in this, data was like a really interesting one, because you can clearly see that this sample from sample 20 to 30 are clearly very different. This was a time course experiment, and these first 15 samples were control, and the next 15 cells, these were the cells, they were treated with insulin, and the last 15 were treated with isoproterenol. And since it was a time course experiment, sorry, the first 5 samples were for 2 minutes, then for 10 minutes and 30 minutes. So as you can see, the labeling here is very different at 10 minutes and 30 minutes, and also different between different treatments. Another example is phosphoenol pyruvate that we saw for the differences here again in the labeling. As you can see in isoproterenol, the labeling we see at C13-2, and in insulin you see at 3, carbon 3 position. So it clearly just gives you, even if you don't, then you can further go on, and you can consult someone here, and if you want to know more about the interpretation, more about biological interpretation, then as Charles mentioned, we do that also. This is just the same figure that I showed before, and that was just by each of the bar graph was by sample, and this is the average values, how these plots look on the whole, and how different they are with respect to each treatment. And it's the same plot. Okay, and so this is pretty much what you're going to get out of the Fluxomics analysis, the Excel spreadsheet, and these plots as a data report. Let's now talk about untargeted data processing. This is the general quality control automation and data acquisition workflow. Most of it is automated. We start by entering the sample information, what we receive from you, all the information we receive, we first input it in our database. Then once the sample is prepped, we also put this information in the database, and then we randomize the sample sequence, and the data acquisition is performed. Once the data acquisition is performed, we sample the raw MS data, and we have a QC standard library, which is basically a list of amino acids and TBOC compounds that we searched against this library for QC standards, and we check after this library search, we check if the quality of the data looks acceptable. Then we proceed the data for then we proceed with data processing. If it doesn't look acceptable, then we go back and perform data acquisition again on the sample. So what we usually look for is RSDs below 30% in these QCs as a standard quality for acceptance. This is a total ion chromatogram, as other people also showed this before, for different samples, and these are different samples, and the x-axis is the acquisition time, or you can call it retention time. So for an untargeted analysis, all the globally all the metabolites that we are looking for are going to appear from somewhere between this 0.5 to 8 minutes of retention time. So this depends upon the type of method that we are using. It can vary depending upon the method that you're using. This is a reversed phase uh, uh, LCMS, reversed phase positive mode chromatogram that you see here. And this plot also shows the reproducibility in terms of chromatography that you see. Each of them, these are different samples, but you see each one of them are aligned just one below the other in terms of retention time. And the intensities, of course, vary depending upon their biological states, but it looks pretty reproducible here. So before we move on further, I would like to just briefly say this term feature, because it's not used globally, and we are using Agilent softwares, so they use this term very frequently, and when we start using it, just gets a little bit confusing. So feature is basically what we call here is a collection of ions at a particular retention time, and it corresponds to a single neutral mass. If you, in a more simple language, you can just call it as a peak. This peak here is a feature. It's corresponding to a single neutral mass, and this is the spectrum for this peak. So this is extracted ion chromatogram, and anything which is like a good peak and has a spectrum like this can be called as feature. So I defined this feature term because we are going to talk about our data processing workflow here. What we start by sampling MS data, and then for each of these these raw data files, we search for features. The feature is just the feature that I showed you. We don't know what it is. We just we run an algorithm in the software which is called find by feature, and we look for all these features in the software, pick up all the peaks, and then store it in our database. So this database also contains the standard library that we have already generated using authentic standards. So we have around 800 authentic standards that were run on LCMS or GCMS and or reversed phase or diamond hydrate on different columns. So in this standard library, we have all these authentic standards with their names and their retention times. So once we find these features, what we do is we store this information along with our standard library information here, and we create another library with these features, and we call it known unknown library. This is known unknown because we really don't have any authentic standards for it, and we just know that these compounds, we see these compounds at a particular retention time. So we combine these known unknown library and our standard library, since we have authentic standards for it, so we know what these are. We combine these things, and then we again sample our MS data in a targeted fashion now, because we For our standard library, we already know what is the mass and the retention time we are looking for. And for known unknown compounds also, we know something is there already present there with a specific mass and the retention time. And we call it recursion, and we use a different algorithm this time to search against the data files, but it is uh, it's a second search in a targeted manner. So once we finish with this recursion, find by molecular formula, we get more confidence in our data because this we run it 2 times and we get more confidence in our data, and then we combine these files, we align them, and we store it in our database. And from our database, Sasha is going to talk more about it tomorrow. We can generate different types of report. Uh, I'm just going to briefly talk of what the basic format of the report. So this is just to show the pathway coverage of our authentic standard library that we have right now. And this shows just a very few compounds that we could map to this pathway graph basically. But we have more than we have quite a few actually. We have around 800 authentic standards in our library right now, and it's also growing. Like we keep adding more and more to this library. So known unknown term, as I use it before, and I just want to define why we call it known unknown. Anything that we know is in our standard library uh, has an authentic standard, but known unknown, the term that we use here is because we have seen these compounds for different data sets. When we run these different data sets, we see these compounds like with same mass appearing at same retention time over and over again in different data sets. But since we don't have authentic standards for these, we don't know what this really is. We can probably guess with their mass and the spectra. We can guess what these compounds are, but a lot of them are known unknowns because we don't have authentic standards for it. But they are equally important because when you do statistical analysis, these guys are the ones that still show up differentially, still show different in differential analysis, very different. So for each of our data set, we generate a separate known unknown library depending upon the data acquisition method. And since there are retention time shifts from batch to batch from different methods also, so for each of the batch that we run or each of the experiment that we run, we generate a separate known unknown library. But all the information of the previous data sets is used in terms of generating this known unknown, and this information we have in our database for all these previous data sets that we have run. So the untargeted report, I think Steve also showed briefly what kind of report you can expect from untargeted analysis. The first, this is just the last column that I've showed here. Basically, you will get a standard QC report. It will be an Excel spreadsheet, so you will get the standard QCs for all the compounds, whether it's known or unknown, and then you will get separate tabs for positive, negative, each mode data. And then this plot here is just the statistical analysis, the final random forest analysis for negative mode data for one of the data sets. and I just wanted to show this because this looks very good, and there's clear separation of the groups here with random forest. So along with the QC reports, with the QC standards and the name of the compound and the peak areas, you can also get this type of basic analysis report with the name list of the compounds which are contributing most to this group differentiation here. Also, Sasha is going to talk about more tomorrow, like what other type of data reports you can generate, or what type of statistical analysis you can see in untargeted analysis. This is pretty much, I just want to thank everybody from my lab for helping me out. Questions?
The random forest that you use, it's not a simple PCA analysis. And why that's used instead of, I mean, it's a form of PCA. Just one of the complaints that I hear from users is that these kind of analysis, you can oftentimes separate groups just because you have so much data, and there's more, there's more data points than there are the numbers of samples that you have. Can you address that question?
I didn't get you what.
Yeah. So because you have so many known unknowns, that you can drive separations of groups often that are more apparent than real because of the way it's done. But what I understand is that random forest actually takes into account some of that the high dimensional data. Could you address that part?
Yeah. You can say that, especially like in this analysis. You can say that the information, this plot is generated in terms of like a PCA plot, as you can see in the separation. So basically, this analysis is people would call it like a little bit more biased analysis in terms of PCA analysis, but it also depends on. Generally, we don't see such clear separations in PC analysis. So random forest, is as compared to PC analysis in terms of this separation is definitely, it's using the same information, and it is a little more biased as compared to PC analysis for looking for just these groups.
So in the report, you get multiple PCs, at least that I've seen it, maybe mine's older, but how many dimensions of a random forest or PCA do you get? It's not just two-dimensional, it's 3 or 4-dimensional?
It depends on the variability. So when you do PCA analysis, it depends on the variability you want to capture out of your data. So for example, a good variability out of data would be, I would say around 70%, depending upon the sample size also. If I want to do, it depends on person to person. If I want to first look of the data, I want to say, I just want to capture 70% of the variability and I don't want to care about rest of it. So I would go and see how many dimensions are capturing those 70% of variation. So if 3 dimensions are capturing that 70% of variation, I would just go for that 70%, just 3 dimensions, and I will give you 3 different plots with each of those dimensions. If my data is not that good, and if 4 dimensions, if I need 4 dimension or 5 dimension to capture that much variation, then I would include more dimension in it to look for that variation. But it depends. So sometimes you can see that variation in just 2 dimensions. You go for at least minimum 3 dimensions, depending upon the variation. But you just look at PC1 and PC2 because the first PCs will capture maximum variation, and you will see clear separation. Other times you have to pick up second and third and then go back and look at your data again with the other dimensions. So it depends upon the variation that you want to capture.
So I just want to take this farther, because these are the kind of reports that people are going to get if they do the target metabolomics. So if you capture most of your variations in your PC1, PC2 plots, then you have a data set that seems to separate the groups the best. Right? Or it tells you have a good data set to separate these groups. Now, what we get is most of these are driven by these unknown metabolomics. But we also get some known. So when a person gets these data sets, how are they going to look at that, of the knowns that are in that data set versus the unknowns? Because the number of metabolites that end up under the first dimension, second dimensions are going to be quite large. And so how do you look at that data, the individual entities that are driving the separation? How do you look at those as far as when they're listed, and how would you recommend people think about that?
So you mean the list of the compounds that we get from the important
compounds that are listed in the order of importance, I guess, for PC1, right? I mean, that's how they're listed.
Yeah. So these ones over here, I think they are based upon the Gini index that we use for random forest analysis. So they are ranked according to that index value for random forest, and they are in the order of their importance. Like, you can say that this is the one that is most important for this separation. So this ranking would be according to that base. So in this list, if I have to say it's from here to here, then maybe this one, based upon just the Gini index, this one is the least important one. So it doesn't basically depend on whether it's known or unknown. Like, I mean, I know what you're saying is mostly that we see the separating compounds are mostly unknown in most of our reports. That's what you're going to see. But yeah, you can, these unknowns, you can go back and look at this mass over here, the neutral mass, and you can search for the compound in any of the database, and you can get an idea what this compound is.
That's going to be addressed by Sasha. Somebody's going to address Sasha.
Thanks, Gary.
Yeah. So that's an important point. So I want to, that's where I was going with this. I want people to understand that you're going to get a lot of, you can separate your groups, but a lot of times you're not going to have a lot of known compounds in those groups. So, sometimes people, at least the feedback I get is sometimes people are frustrated by not having, identified compounds that you can say this is the biology that underlies this. And so, but there is information there that we can go back and do it. Now, go ahead.
If I may comment on that. So, I think I know very well what you're getting at. And so, I think the useful way to think about this particular view in the report that you might, in untargeted report that you might get. I mean, you're not looking at the biology yet at this point, okay? So you're not ready to really identify which molecules are, really significant and maybe sort of, related to particular changes that you're looking for in your experiment versus your control. This is just sort of your first look at the data. Okay, so you're getting an overview of the data. What kind of information can you get out of this report? Like Tanu said, this is really, we're seeing really good group separation here. But the thing to keep in mind is, that's what Chuck said also repeatedly, okay, there are a lot more variables than samples at this point, okay? So the thing to be aware of, and many of you know it, I'm sure, is overfitting, right? And that's a typical situation when you have more variables than samples, okay? Then sometimes you may just, get suspicious if you see some very, very clear separation between groups at this point. I mean, you have to be careful that it is not driven by, any particular feature, set of features, okay? And then the thing to do maybe is exclude that from your further analysis. But the things like that, I mean, I think can be clearly, sometimes clearly seen from this type of overview, okay? So I think that, hopefully tomorrow we're going to talk more about different steps in the analysis. This is really the very, very first view of the data, whether it's done by random forest or by PCA. And I think what Tanu and Sasha are leaning towards, or saying that random forests seem to be a little bit more robust for doing this type of thing. Is that right? Or PCA? I mean, I don't know.
I mean, it depends.
I guess, right, sure.
It depends on the data, and what we're trying to say here is, like, tomorrow Sasha is going to talk more about, how he's going to talk about his tool where, where clients can log in and look at their own data and can look at random forest. At the same time, they can look at PCA, and you can create your own reports and see which one you want to look at.